Afternoon, everybody. I'm Ann Barker. I'm co-director of Complex Adaptive Systems at Arizona State University and director of the National Biomarker Development Alliance, and I'm a professor here at ASU. It's a great year to be talking about biomarkers, and I'll come back to that in a second. But the title of this talk is Biomarkers, an Auspicious Beginning and a Portentous Future. And I'll say more about that as I go through the talk. 3 points to think about before we start. One is there's an opportunity on your screen for questions and answers. And so if you have questions, just write them down and we'll attempt to answer them. If we don't get them all answered, or if you have questions after the talk, you're welcome to submit those. The second thing, this talk is being sponsored by Roche, and we appreciate their participation. And last, this talk is also eligible for CME credit. And if there are any questions about that, you can contact your institution. So let's get started. We're going to talk about several things today relative to biomarkers. It's a space where a lot of things are happening and a lot of things are going to happen. So I'm going to talk today about a few things that I'd like to bring to your attention, and one or two things that I think will be critical for our communities to start thinking about. One is we're going to start with some definitions. First of all, I don't know that anybody actually has a great definition for biomarker, and if you ask 10 people, you probably will get 10 different responses. But just for the purposes of this talk, we're going to define biomarkers in a fairly classic way. I also want to talk a little bit about the beginning of the biomarker science space and how that's kind of shaped what we're doing today, and how it's tended to shape maybe what we'll do in the future, but we have opportunities to change that. The other thing I want to talk about is the state of the science of biomarkers, and it is a science. As a matter of fact, it's probably the largest. It's probably one of the largest spaces in the literature field today, and we'll come back to that. It's presenting us some really tough challenges, and we're going to have to deal with those challenges as a community. If you look to the future, we're faced with some really, really interesting and sometimes daunting problems, and it has to do with the complexity of cancer and the difficulty we have with capturing that relative to what a biomarker is and what a biomarker might be in the future. And in recognition of that, I'm going to bring to your attention some concepts that FDA has embraced, some new regulatory paths or some updated regulatory paths that need some real interest from our communities in terms of the help that we need to give the FDA to build the evidentiary standards for biomarkers. And then last, I'll tell you about a consortium that we built in the last couple of years, the National Biomarker Development Alliance. It's a trans-sector model that everyone's welcome to join, but this is probably the model, this is probably the way we'll build these evidentiary standards in the future for biomarkers. And so with that, let's get started. So what are we having, why are we having all these meetings and talking about cancer, and any given week you can go to, I don't know, you can pick a number of biomarker meetings. It's because of the burden of this disease. And I always start with a slide like this because I spent a long time at the National Cancer Institute, and I always started out by saying this is a daunting problem. It's one that actually consumes a lot of our resources, but it also consumes a lot of our lives, and it takes a lot of people's lives. And if you think about these statistics, and you can read them there, 25% of our population will succumb to this disease. And as our population is aging, we're talking more and more about this now, 77% of cancer is diagnosed in individuals over the age of 55, which means that cancer in our country is going to go up anywhere between 30 and 40%, in as little as 15 to 20 years. That we're already on that curve. And if you look at the cost of cancer, it's certainly in the hundreds of billions of dollars. At NCI, we estimated it to be somewhere over 200 billion. That's just the direct cost of this disease, not the cost of lives lost and productivity lost from losing those individuals. And it is not just the U.S. problem. If you think about the problem globally, it's becoming one of the major issues of this century. And the World Health Organization, just I think 2 years ago, came out with these projections that by 2020, there will be 10.3 million deaths and 16 million new cases of cancer diagnosed every year globally. And so that's a 70% increase over the next 20 years. Those projections were in 2014. When I was at the NCI, we had lots of conversations with lots of countries trying to prepare for this. Countries like China, for example, where the population is absolutely enormous, 350 million smokers in China. Again, we have no biomarkers to predict which of those individuals will get cancer. So on a global basis, the economic impact and the human impact is going to be staggering. So one of the tools that can actually help us to impact this is going to be biomarkers. So if you look at the biggest problem we have in cancer today, and we are making some progress, but not nearly enough, it's metastatic disease. So if you look at the concept of a primary tumor where there's no metastatic disease versus one where you have metastatic disease or you develop metastatic disease, with most of these cancers, we don't do very well. We have never done very well. And this is another area where biomarkers are going to have to come into play and help us to begin to predict these individuals that are either move on to metastatic disease or to predict essentially when recurrence is going to occur after primary treatment, et cetera. So the two really daunting problems that I think we're facing is one, that just the global incidence of this disease, and two is the complexity and nature of this disease, most especially metastatic disease. So that's really brought us to the cusp of what we're all talking about today, and this has been preceded by a molecular biology revolution and a lot of advances in molecular sciences. But essentially our vision now for 21st century medicine is precision medicine. And you see it every day now. People are getting their genes sequenced, their genome sequenced. This is going to physicians. They're actually starting to really use this as a tool to treat patients. The obvious problem is the number of limitations we have on the number of drugs that match those signatures or those biomarkers. But the healthcare realities are coming back to our previous thinking on the global nature of this disease and the economic impact. This is one of the major issues with healthcare today in terms of cost. And spending in 2013 was $3 trillion. It's going to continue to rise at 4% or more per year to become 20% of our economy. Our dream for the future is not essentially to treat established disease, not to use cytotoxic therapies, not to have people diagnosed extraordinarily late, but to have an entirely new approach where biomarkers can drive us to detect disease early and actually treat it in a targeted way, knowing what the molecular profile is. And that connects patient care back to the laboratory in a continuous cycle, and that is all dependent. Precision medicine is dependent on the availability of robust, very, very effective biomarkers. And that, in fact, makes biomarkers probably the highest value in precision medicine or any kind of medicine today. So the highest value proposition is probably going to be biomarkers. So what are we talking about? This, like all things, we have defined this whole concept of biomarkers quite officially. And so you've read this definition probably numerous times, but a biomarker is a characteristic that's objectively measured. That's very important. And it's evaluated, usually a change in the patient that you can measure. Is an indication of a normal process or one that represents pathology of some kind or something you can measure after you intervene with a drug, say, or a biologic. And the small print is very important. It should be clinically relevant, fit for purpose, which means it should be used and useful for exactly what you say you're going to do with it. It should be measurable and reproducible, big issue with biomarkers, and directly reflect the state, either the normal or the altered state. That's a lot of things to think about when you start defining these things. So one of the really important uses for biomarkers is as surrogate endpoints. Now, if you think about this, this is kind of the holy grail of biomarkers. And essentially, this was defined quite a long time ago by Robert Temple at the FDA. And it hasn't changed, essentially, in terms of what a surrogate endpoint can be. It's a laboratory measurement or physical sign that's used in therapeutic trials as a substitute for a clinically meaningful endpoint. And it has to be a direct measure of how a patient feels, functions, or survives, and that's expected to predict the effect of the therapy. Now that, if you put all that together, it means that biomarkers as surrogate endpoints are pretty rare. And in oncology, they're almost non-existent. So the idea that we can develop biomarkers that can substitute for clinical endpoints is a terrific idea. And it's actually the basis for accelerated approval of the drug at the FDA. But to this point in time, we have very few of these for some reasons that we'll talk about as we go through the rest of these slides. Some other terms that I like people to remember when you're talking about biomarkers: you have to build assays for them, obviously. You have to know what sensitivity is. You have to be able to correctly identify, essentially, the biomarker that you're looking for, the specificity. The precision is the closeness between the independent results obtained under these defined conditions over and over again. Fit for purpose essentially is used by the FDA. It defines the context of use for the biomarker. That's a very important term. And then receiver operating curves, anybody who works in this field essentially uses these to determine the accuracy of tests. So these are, if these are not terms that are familiar to you now, and you want to be in the biomarker space, these are terms that will become familiar. So biomarkers are, as I said, probably the highest value that we have in the whole field of precision medicine today. That's not to say that you can't, if you got a blockbuster drug, you can bring in a lot of revenue. But if you actually think about what you could do if we could really, really build the right biomarkers in terms of the value of biomarkers and the value proposition, it would be incredible. It'd be incredible because you could drive therapy development, detect disease very early, help you choose treatments, assess whether or not your treatment's working, give you a good prognostic indicator so you can tell your patient what to expect. Essentially, we hope someday to prevent with biomarkers, understand what kind of changes might be important in terms of disease onset. Then treatment, disease monitoring would essentially occur. Treatment disease, sorry, my screen said that we had disconnected. I hope that's not true. Treatment, disease monitoring, surrogate endpoints, essentially, as I said, and accelerated approval for a drug that essentially where you have that kind of endpoint. So if you're a patient, you might ask some of these kinds of questions. For example, what type of cancer do I have? Or is this the right drug for me? Or what's the optimal dose of this drug for me? Or will the cancer return, et cetera? So this is also an area where patients will become more and more involved because as they know which questions to ask, biomarkers will become much more patient-centric. So let's now talk a little bit about this auspicious beginning for biomarkers. And biomarkers go back almost as far as one can imagine, because a biomarker can be nearly anything in terms of the kinds of things that you can measure. It could be an image, so imaging biomarkers are very powerful. It can be some of the early kinds of biomarkers that I'm going to tell you a little bit about now. It can be these very sophisticated biomarkers. It can be algorithms that integrate big data sets. Everything's on the table when it comes to biomarkers. If it meets that criteria of measuring that sort of change and doing it reproducibly and measurably, it can be a biomarker. So some early wins. So there were a lot of early wins in the biomarker space. And now we're talking about very early. We're talking about last century, probably as early as the early 1990s, you started to get biomarkers approved. Cardiovascular disease led the way, and everybody's wanted to look like cardiovascular disease ever since. And so we keep talking about these great biomarkers, like blood pressure, and that's one that still is used remarkably well for all kinds of drug development. HIV, this was actually prompted by a crisis, but CD4 counts are used every day as essentially in drug development and also in patient care. And then in diabetes, hemoglobin A1C, you'll see it coming up all the time with FDA in terms of how drugs are assessed and used. So I want to tell you a little bit about this cardiovascular story because it's a really complicated story, and it's one that we can learn from. So I'm going to tell you the good news and the bad news here. So surrogate endpoints, and this is the disease that has the most surrogate endpoints when you think about it. This is a really good case for successful approaches. And I'm going to flip down the slide a little bit because this all started with the Framingham study, which is a huge cardiovascular study sponsored by the NIH, started as early as 1948 at NHLBI. And essentially it was to sort of find out what causes heart attacks and stroke. It had more than 5,000 patients, I think by the time they actually were done. It was published in 1961, and they actually isolated and actually had sound data for these risk factors, like cardiovascular disease and cholesterol levels and blood pressure, et cetera. And even in some of the arrhythmias, we were measuring by ECG then what those arrhythmias were and what they might mean. So these things became the targets very quickly for drug intervention. And in combination with all kinds of other technologies, imaging, it's produced really profound results. The decrease in mortality from cardiovascular disease has been stunning. And as a matter of fact, all-cause mortality, the biggest impact probably has been from this whole issue starting all the way back in the early part of the last century, that we actually did a study that really changed this disease and changed all-cause mortality. So this really prompted an enormous number of clinical research studies. And if you think about, you think about the fact that all these biomarkers have been approved and been in place for many, many decades now. The clinical trials, which were really quite early, started in the early 2000s, were really quite seminal in setting sort of the course for how biomarkers would be developed and approved. So if you look then at how many of these things were so beautifully done and were impacting very positively, there was an event in this whole process that changed just about everything in the way we develop biomarkers and the way we judge them and how careful we have to be, especially around surrogate endpoints. And this saga essentially is best captured in something called the CAST trial. And essentially the CAST trial was based on the concept that ventricular premature beats was an excellent surrogate endpoint for survival after an MI. So, and this was, at this time, sudden death after an MI was, at least the physicians felt as though it was very tightly correlated to these premature ventricular beats. Things are not always as they seem. So the CAST trial began in 1980, and there were just a slew of arrhythmics developed during that period, and because it was a very popular sort of class of drugs, because it was going to essentially make a lot of revenue for the companies that were making them. So in 1980, it's very interesting. It was so accepted by the community that this particular biomarker was in fact a surrogate endpoint that people didn't really want to do this trial. They thought it would be unethical to do this trial. But anyway, science persevered, and they did the trial starting in 1980. And they looked at the effect of these 3 drugs on in post-MI in a lot of patients. And essentially, their premise was that this endpoint would actually be predictive for survival in these patients post-MI. So they were able to suppress the VPBs nicely, probably by half, I think, but they doubled mortality. So essentially, these arrhythmic drugs increased mortality significantly. They were essentially wrong. The whole premise was wrong. It cost a lot of lives. So what people finally internalized is that surrogate endpoints have to think about or have to consider the fact that you are going to have to integrate a lot of issues if you're going to come to a clinical endpoint. So this was the first real discussion of off-target effects and other factors that come into surrogate endpoints. And it was consciousness raising at every level and essentially made our communities understand once and for all that surrogate endpoints are extraordinarily difficult to achieve, and biomarkers achieving that status is a very, very high bar. If you look at what happened in cancer, and this started this particular drug, Gleevec, which is in fact the drug that started the whole revolution in cancer. Around, again, probably the mid-2000s, people began to work on this. Brian Drucker at Oregon Health Science University is responsible for the development of this biomarker. And essentially, this one was discovered in a way that took about 50 years. It was observed 50 years ago that the Philadelphia chromosome has, that all of these patients had a similar sort of genetic defect, but it took another 50 years to figure out where it was on the chromosome, how it behaved, et cetera, and to get it modeled. But this is sort of the way that I think the field has gone since. We really fell in love with the idea that if you know a molecular change in a tumor, then you can probably build a drug to it. Now, that doesn't really consider, in some cases today, that this particular molecular change has to be totally related, or certainly in principle has to be totally related to the disease and the causation of the disease. This is true for chronic myeloid leukemia and this particular abnormality. It was almost a miracle drug, and it was approved in 2002, probably one of the shortest review cycles in the history of the FDA. So you have 2 stories played out here, really 3. One, you have the very successful early days of the cardiovascular world, where you had things like blood pressure and cholesterol and some of these other biomarkers that are still used today, and it's really affected, really affected mortality. Then you had the CAS trial, which has got everybody's attention in terms of what a biomarker should be and what a surrogate endpoint, a biomarker that's going to be a surrogate endpoint, has to really be. And then along comes all of this progress we have in cancer, and we turn our attention to molecular biomarkers. And this, along with HER2 and Herceptin, are one of the 2 kinds of biomarkers that have changed the field forever. So we started out with a very audacious beginning, and we have a very portentous future because of our molecular sciences and the kinds of projects that we've done, things like the Cancer Genome Atlas, et cetera. It's giving us a lot of molecular data. So let's look at the biomarker field. Interesting, the biomarker field, like most of cancer research, is driven by technology. And I don't mean that in a way that says it's not also driven by intellect and team science and innovation and all the other things that we think about. But what we use every day in these laboratories to create biomarkers are really technologies. And those technologies keep evolving. And it started out a long time ago with very simple kinds of approaches to sequencing, which is called Sanger sequencing, still use it today. We now know how to look at the transcriptome very carefully through RNA-seq. Our mass spec is we still have issues with some of the standards, but it's becoming a very powerful technology. Epigenomics has found its way into most people's hands now. And we're beginning now to think down here in terms of biomarkers and the way that cells function, communicate, etc., is going to be also closely tied to the spatial and microenvironmental effects. So there is coming out of all of this a multidimensional data explosion that's amazing. And if you look at this, this is essentially what's also driving biomarker science, huge amounts of data. And that data is coming in all kinds of forms. The variety is amazing when you consider the clinical data that has to go with all molecular data. The rate is unbelievably almost exponential now in terms of acceleration. And the data types are pretty much almost unbelievable in terms of the numbers of types of data that we're getting today. So that is producing huge opportunity in our field, but it's also producing huge challenges. This, if you talk about a portentous future, this whole issue of big data and how we're going to analyze it, how we're going to turn this into biomarkers for understanding the natural history of disease, for actually coming up with biomarkers that we can approve incrementally up toward becoming, say, a surrogate endpoint, is probably one of the greatest opportunities we've ever had in biomedicine. So what's driving all this? I had something to do with some of this. Dr. Collins and I, when I was at the NIH, NCI, we started something called the Cancer Genome Atlas, which has produced petabytes of data on many, many tumors. But these technologies are now essentially driving almost unimaginable amounts of data actually being developed. And globally, in 2014, I think it actually exceeded 28 petabytes of data. And nobody quite knows what this is going to look like in the future, but there are countries actually proposing to sequence huge segments of their population. And 1,000 petabytes a year probably is not an unreasonable way of thinking if, in fact, this is going to be the foundational element of precision medicine. So how do you understand all this? How do you bring any kind of understanding to that much data? So here's an example from the Cancer Genome Atlas. This is the first paper we published out of the Atlas. And it was interesting because it allowed us to sort of think this through. How do you organize data? So we organized this data around known pathways and pathways that were very important, especially in the glioblastoma process. This is a slide is from the glioblastoma paper. But essentially, what we found, the further we looked, is yes, we could subtype these different diseases within GBM related to their molecular changes. But if you looked at it on a patient-by-patient basis, they were more like snowflakes, essentially. I like that term because there was an organizing construct that we could look at, but within the organizing construct, every single one was different. And there is a real message there that we have to understand in terms of how we use genomic data in the future, because every patient's going to have a signature. It's going to be part of a larger signature, but every patient's going to have their own unique signature. So I want to say a couple of words here. I gave a talk on this a couple of years ago, maybe here, I can't recall, but I talk a lot about this because the National Biomarker Development Alliance is trying to attack this problem and a lot of the other problems in biomarker science. But I'm going to stop here and talk a little bit about this because this whole data explosion, which has been going on, it's been going on for almost 30 years now, maybe longer, certainly since we finished the completion, or we completed the human genome. But it's produced an embarrassment of, we thought, riches, but in fact, we've got literally hundreds of thousands of biomarkers published in the literature. Literally, if you go look, you will be stunned. And this has become a real focus for a lot of the public, and increasingly for the Congress and other people who think about the value of research. And even today, there's an article in Christian Monitor, the Christian Science Monitor today, on the reproducibility of science. And it's been covered in Science several times. It's been covered in the New Yorker. It's been covered in The Economist. It's been covered in, you know, in Nature. It's been covered in almost everywhere, in every public sort of periodical. It's the reproducibility of these data is important because, as I've just shown you, unless your biomarkers are robust, sound, it's going, and other people can reproduce them, it's going to lead to this kind of problem. And so this is a problem that communities have to address to make biomarker science the science that we need for the future. That portentous future will only be realized if we solve this biomarker reproducibility problem. And just to show you, this is my colleague George Post published this paper. I always like to show it because this was all the way back in 2011, but 150,000 claimed biomarkers that year, and essentially we've approved about 1 protein biomarker per year since the early 1990s. So it's a pretty dismal picture. But there's great opportunity here. I'm not very pessimistic about it. I think all these problems can be solved. So what we're going to do now is we're going to talk about that a little bit. So how do we take the sort of the audacious beginning of the biomarker revolution? We learned a lot from cardiovascular disease. We learned a great deal from the way the BCR-ABL, for example, alteration was developed and that drug was developed. Same thing with HER2, and we've had a lot of those, not a lot, we've had some of those examples since then. But now we're looking at a future, and it's very portentous with promise, but how are we going to actually navigate the future to get there? And what has to be done? What do we have to think about? What do you have to think about in your laboratory? So if I look to the future, I think the one issue that's going to really be important that we understand and we be able to start thinking about how you capture real complexity of cancer in something that we call a biomarker. And that means that a lot of us have to change. So this data tsunami that we're seeing, we've got to learn how to use that in our favor, essentially, and use this to reproduce data, and also to open new doors for how you develop, not just how you discover biomarkers, but how you develop them. So I think we should embrace this period, because I think it was an author wrote recently in Science, sorry, I've forgotten his name, but he mentioned that this whole crisis in reproducibility is really a huge opportunity for our field. It's happened in other fields, and going from, from sort of a prototype to manufacturing, or all the data that goes into a prototype before you get to manufacturing. Other fields have had to go through this, and we're going to have to go through it now, and we can. So this is a slide I made a long time ago, and I still like it. But essentially, cancer is a complex, evolving system. It's not just a system that you put in place and it never changes. It's evolving, and all the environmental effects that you have internally and externally can impact this system. So if you think about this, what you've got to be able to capture is, once you get out here the malignant state, you've got to be able to capture that state. And the same thing's true with earlier states. It's a challenge, but I think it's going to be doable. So if you think about it as a complex adaptive system, a lot of the hallmarks of cancer kind of come into focus then, because you can see the interactions as to why, for example, angiogenesis is an important aspect of cancer, or its ability to replicate uncontrollably is one of the hallmarks. But essentially, the numbers of elements, which are multiscalar, the robustness of the system, a lot of backup systems in cancer, it's very adaptable and very evolvable. And that, in fact, leads to what we think of as the metastatic phenotype. And that produces all kinds of problems for us in terms of resistance. So we have to understand this disease much better as a complex system. So how can you do that? I don't have the answer to this. I mean, I have a lot of ideas, but one thing that I'm intrigued with is exosomes. Exosomes are really interesting in the sense that they capture a lot of complexity, and they can also capture some aspects of the microenvironment as well as some of the evolutionary kinds of aspects that we're looking for. So I think this is one of those areas that will have some real promise. I think also another area that I like a lot is this whole issue of algorithms and how you develop algorithms to really interrogate a data set that will tell you something about the question you're asking clinically. And I see a future where we will have algorithms serving as biomarkers. And there are others. I think circulating tumor cells has promise. We haven't actually been able to do a lot with that promise yet, but there are aspects that I think we should start thinking about that are much more complicated than those single genomic changes or even a few genomic changes. We're really early in this revolution, and we need to all understand that. We've got a long way to go. So I want to talk, I wanted to come back to surrogate endpoints because I think that if we're going to eventually replace many of these very long clinical endpoints, which require sometimes years to actually get to survival or another clinical endpoint, then I think we do, coming back to this complexity question, how do we actually start to think about surrogate endpoints? And there are some long-standing issues here, and I wanted to bring them to your attention today so you can begin to think about them as you start to think about your own career in the biomarker space. So coming back to what we said, this line that I've underlined here is in fact sort of the key issue here. It's how a patient feels, functions, and survives is a very interesting way to look at this. And there are lots of examples where people have submitted very, very sophisticated biomarkers for certain diseases. And if you think about it, something very practical turns out to be a much better biomarker because it's a measure of how the patient is functioning. So biomarkers actually are classically defined mathematically. And this particular Ross Prentice, who when he wrote this was at the Hutch out in Seattle, but essentially this is the criteria for a surrogate endpoint to be a surrogate endpoint. So if you think about this, it's a true endpoint, that's the clinical endpoint. The surrogate endpoint is what you hope that you'll get approved. And then Z is the variable for treatment. And the effects here and the interactions here are really quite profound and extraordinarily difficult. These criteria have never been met for a surrogate endpoint. So I'll just leave that. That's the ideal. There have been surrogate endpoints approved, but not that met this criteria. So here are some surrogate endpoints that we all in cancer know a lot about. Pathologic complete response. Essentially, we got that approved a couple of years ago for neoadjuvant breast cancer. Time to progression is in cancer is a valuable surrogate endpoint. BCR-ABL I mentioned before, and these are a couple of the ones that we know from other fields, but essentially glaucoma and cardiovascular disease represent two very strong surrogate endpoints. So there are some huge challenges here. So what is it about biomarkers that makes all this so difficult? It's the complexity, coming back to the complexity of the disease. So it is unlikely that a single biomarker, even maybe a few genes, are going to tell you all that much about the clinical outcome. It's a multifactorial, multi-systems, organ-based, and the organs are connected and variable in their responses, and the effects of drugs on those organs can be very profound. So one clinical endpoint may not provide the complete picture for the overall clinical outcome, because multiple clinical endpoints can even they don't necessarily even correlate with each other. So the clinical endpoints themselves can be an issue. The drug or the biologic may do other things, as was showed in the CAST trial. You may in fact have all kinds of off-target effects. And then surrogates may not be related to outcome, again, as we saw in the CAS trial. And it turns out that, you know, that is an endpoint. It's just not a clinical endpoint in the CAS trial. It doesn't predict the clinical outcome. So another example is a white count or fever, essentially. Those are very good examples that are not related to the clinical outcome of the patient. So I'm going to stop here and give you some information that I want you to take away from this talk, and that is most people are looking to biomarkers to be surrogate endpoints, essentially to predict what's going to happen in a patient, exactly what's going to happen in a patient. That's a very high bar, very high bar. There are lower bars that will allow you to use biomarkers in an extraordinarily high value way. And I want to tell you a little bit about that, because I think the surrogate endpoint story has taken us away from some of the real promise of biomarkers that can be used in patients to do a lot of things that need to be done. So biomarker qualification is essentially defining a context of use for a biomarker and then figuring out how you qualify that biomarker. So if you think about the whole process of biomarker qualification, it's not exactly like drug development, but it's a little bit analogous. So essentially, you've got to decide first on the context of use. You have to decide on a use statement. In other words, how are you going to use it within that context? And then what are the conditions for qualified use? So you have to be pretty concise in all of this. You have to think it through, and you have to have evidence. So if you think about the process, and just let me just take you quickly through the process. So biomarker qualification is really helping us to develop a whole set of tools to do a lot of things, to develop drugs most especially, but other things as well. So if you think about it, safety and effectiveness, assigning patients to clinical trials, does the patient have stable disease? There are all kinds of questions that you want to ask in a clinical trial, and you can do that. So here's the process. FDA has started this process all the way back in 2004, revised it. This slide that you're looking at here is from Dr. Federico Goodsed, who started that process at FDA. But essentially, what you do is you or your organization can submit an application through a letter of intent to the FDA. And then essentially you're going to have to say what you want to qualify and what the context of use for that is. That will go into the FDA and get reviewed. And then you will present to the FDA a package that contains your context of use and all of the evidentiary standards that you need to prove that context of use. And once FDA approves it, they put it into the Federal Register, and essentially, once they get their comments back, it can be used. And what's really interesting is that once it's approved, other people can use it. It doesn't have to be reapproved. And it's independent of the technology. So it's a very powerful approach to actually doing the right things with biomarkers with patients. And I personally am a big fan of this process, and it's gotten a lot of attention this year because of the 21st Century Cures Bill, which has been rolling around Capitol Hill now, like most things. But there's a very big section in there on how you actually do this. And so the one thing that I'll come back to in a second is FDA is not going to develop the evidentiary standards for these various contexts of use. The communities have to do that. It's going to require consortia to do that. And this is a place where our communities can start to take leadership. So I'm going to give you an example. Okay, the best way to do this is just to show you how you might do this. So again, giving credit to my colleague, Dr. Goodsed, he believes, and I think he's right, that if we do this right and do it well, we can start to develop the evidentiary standards as communities in an incremental way. In other words, you can develop your context of use incrementally. Everything can't be a surrogate endpoint. So maybe you start with a lower level of context of use and you work your way up in terms of standards. And here's an example for circulating tumor cells. Now this is all kind of made up, but let's just say that you wanted to select patients for clinical trials. And essentially, the action in terms of what you have to do for CTCs, there is an approved device. What you would have to do essentially is not very much, if anything, to essentially say that you could use this technology or use this biomarker. But essentially, you want to then lay out the risks and the benefits, and then you want to define the evidentiary standards that are needed. So here, let's say you need to establish the correlation of CTCs in a prior experiment and number prior to initiation of therapy and survival and prognosis. So you've already done that study. So this might be an approvable context of use. This first line might very well be approvable. If you look down at the bottom, avoiding futile treatments, for example, that would be a biomarker. The context of use would be treatment resistance. So all of these things can be sorted. If you know what context of use, you understand what the risk is to the patient, but what the benefit is to the patient, and you can define the evidentiary standards. You can present that to the FDA. That's a very powerful way to go to the FDA to get your biomarker in your hands to use in the clinic in a way that will benefit patients. And it doesn't all, and by the way, these are not diagnostics, nor are they companion diagnostics. So you have to understand that these are tools that can be used in clinical development and certainly in drug development. And I think they're probably one of the biggest, maybe the most promising area we have in biomedicine today. To show you a little bit about maybe how you could approach this, if you're interested in sort of how you fix these problems, let me give you just a little history of something that I did over the last 2.5, 3 years. I am extraordinarily frustrated with the state of the biomarker science everywhere, and did a lot about that. Tried to do a lot about that when I was at the NCI. We spent a lot of time, funded a lot of projects, especially National Initiative in Proteomics. We did the same thing certainly in genomics. We tried to bring real rigor to the way we identified genomic changes through the Cancer Genome Atlas, et cetera. But the truth is, biomarkers, that's probably the largest segment of the data that is so being, blasted out there as being not reproducible. A lot of that's biomarker data. And we aren't approving biomarkers very easily because we haven't done the work. We just haven't done the work. So if I look at what we have tried to address, essentially is one of the things that Janet Woodcock said years ago when she developed the Critical Path Initiative at the FDA, is that we don't have any common standards or best practices or guidelines to drive end-to-end biomarker development. And it's a process that starts with discovery and ends when you get it approved, either as a surrogate endpoint or as a development tool if that's your ultimate goal. But we don't have any real decision tools to go through this process. And it's not FDA's job to develop these standards, it's our job. And so we took that on at the National Biomarker Development Alliance, and we've been working on that for a couple of years, almost 3 years, with a couple of hundred experts. And we've had a lot of success. But our overall goal is to develop some systems that the communities can use to bring solutions to their work so that biomarker discovery can move through development and into patients at a much more rapid rate. And we can actually achieve precision medicine. So coming back to, sorry, that's the wrong way. This is some of the work that we've done. We hold these think tanks. And if you want to join us sometime, it would be, it's really fun and sometimes painful because we deal with tough problems, but we've tackled everything from biomarker discovery to the surrogate endpoint to some of the issues with clinical trials and development itself. In other words, how do you actually go about developing a biomarker? And all of these things are available for you to review on our website. But essentially, everything we did has taught us a few things, and there are things that you should take away from this talk. Every biomarker experiment has to begin with a clinically important clinical question. If you don't start there, you end up at the end of whatever it is you're doing, potentially with an answer, but an answer that has no real relevance. So you have to have a robust experimental design. That means you have to talk to statisticians, and you have to be sure that the statistics is going to support the answer to your question. So much of our data is not statistically sound. Biospecimens, one of the things that I have worked on just for a very long time, and the biospecimens here have to be, they really have to be robust in terms of quality. You have to have the right numbers depending on your statisticians. The technologies that you employ have to use standards. You have to use standards. And it's not that we don't have standards for technologies, it's we have all kinds of standards. And then all the data that you collect must be of very high quality, and you must have the provenance with the data or the metadata to tell you where the data came from. And then finally, this is a weak area. Analytics and analysis that are responsive to your experimental design, they must be appropriate and robust. And this is an area where we need a lot of training, because in looking at, say, expression, looking at the transcriptome, et cetera, you've got to be able to go in and use the right kind of algorithms to find signal in some of those data sets, and then reproducibly go to another data set to see, a random data set, to see how valid your signal is. So we came up with this little graphic, and essentially this is not this is not the way the world works. I mean, some of these things go on obviously in parallel, but early discovery is where, or discovery per se, is where most things start to fail. And making a decision to translate something from that red circle to that blue circle involves a lot of money, and it should be an extraordinarily carefully made decision, because once you start building assays and you have to develop them, then you have to know if this assay is going to be scalable, it can answer your question, it's met all those other criteria. And then the purple circle there is qualification. If you qualify your biomarker, you can use it in all kinds of ways to build the best clinical trials, to actually find out about the toxicity of your drugs, to dose drugs better, et cetera. And then somewhere out here on the yellow, in that yellow circle, you can actually turn these things into surrogate endpoints by validating them in clinical trials. And standards are required at each and every step of this, and that's where we're falling down. I think that's going to happen. It is happening now. Lots of groups like ours are really getting on board in terms of developing these kinds of standards. So we asked ourselves a few questions. We said, are there systemic problems here that we can address? And our answer was yes, there are some systemic problems. So we've taken on one. We've taken on two. I'll tell you about the second one. But this one is very, very interesting. In talking to our chief medical officer at the NBDA, Dr. Carolyn Compton, she said, you're never going to be able to do precision medicine in the communities because the pathology labs are not set up to do it. And they're overseen by the College of American Pathologists, and it's just, it doesn't fit in the hospital flow, and all the things you want to do to have the quality that you need for molecular assays is extraordinarily difficult to implement. So we formed a partnership with the College of American Pathology. We had a couple of workshops with them, and essentially what they did was to tackle this problem very interestingly. They said, what are the 80% of the effects that cause failure of biospecimens? About 20% of them, actually. So they came up with a list of, of variables that have to be controlled, essentially, to get from any hospital, anywhere that CAP oversees around the country, what do they have to do to produce a very high quality biospecimen? So those performance metrics are being developed, and CAP and NBDA are working on a process to actually institute those. And that means that eventually any patient can get a very high quality molecular assay in any hospital if CAMP is actually overseeing the implementation of these particular performance metrics. Because if 80% of the problems come from 20%, that's all fixable. So some of the other things that we're working on, essentially for this whole issue of how do you come up with a good clinical question, we're trying to establish some baseline biomarkers for certain diseases and outcome biomarkers for some other diseases, or those some of the same diseases, and looking at biomarkers for some of the stochastic effects of treatment to help us to begin to help others to define what is a clinically important question. And then design-wise, this is tough because a lot of people aren't really experts in design. So we're in the process of trying to put together a commons for biomarker design where we could put some of these models up for people to understand. And as I said, we've set up this approach with the College of American Pathology to try to control the quality of biospecimens. And we're undertaking with the FDA some case studies for biomarker qualifications, 4 of them actually, to say that here are 4 diseases. Here's what we believe the evidentiary standards would have to be for these contexts of use. Here are the risks, et cetera. So these case studies will be very important. And if you have a case study and you'd like the NBDA to work with your case study, then just contact me. So we're not a big organization. We're a few committed people who actually have a lot of committed colleagues. So we're a small organization that leverages an enormous amount of expertise and have a lot of progress because of that. And this is the future. I think this is the way the world is going to be. I think a few people can leverage a lot of the intellect that's out there to solve these problems. So maybe a few years ago, I saw this slide. Danny Ellis, who worked in one of the programs I had at the NCI called the Physical Sciences Oncology Centers, quite an accomplished mathematician. So he said, maybe this is what we should be thinking about in terms of the way we're going to treat patients in the future. Since patients are very complex systems, you have to start thinking about this as the inputs and the outputs, and then how you're going to actually manage those. And biomarkers is a big piece of that. The molecular data is a big piece. But there are all these other biomarkers and other outputs that you're going to have to consider, and especially what comes from the patient. You're hearing a lot today about patient-reported outcomes. So what's the patient saying about the drug? And all of those things would feed into a lot of information, a very high information content set of interventions that would be used in the future to actually treat patients as complex systems, which I think biomarkers will be absolutely pivotal to achieve. But I can begin to see that portentous future. I can begin to see that we can fix these biomarker problems and potentially offer this kind of an approach to patients, which would probably be comprehensive and would do a great deal more in terms of increasing longevity and quality of life for patients than we're doing now. So to summarize, I've given you a lot of information today. What a biomarker is, what a surrogate endpoint is, what we mean by context of use, the approaches that you can actually take through the FDA to get your biomarker into some kind of development process. I favor the qualification process. It's an incremental process. It's one that requires evidentiary standards, but we can, as a community, we can fix that. And it's up to you to organize your own. If you've got a context of use, if you've got a group of people who want to work on that and want to do a case study, then I would say that's the way to start. Finally, those of you that know me, I believe very strongly coalitions of the willing are the way to change the world. And for this biomarker issue, and there are many issues, I think this is something that will depend on the community's engagement in developing those evidentiary standards. And I know FDA is depending on it. So I thank you for your time today. We don't have much time left. I'm sorry, I had a long presentation. But if you have any questions, you can type them now, or you can send them after this is over, or you can send them to me, my email address is Anna, A-N-N-A dot Barker, B-A-R-K-E-R, at asu.edu. And I would especially love to hear from people who have great ideas for biomarker qualification cases, because I think the FDA is going to have a number of those submitted over the next few years to really begin to build these processes so we can get these biomarkers into clinical trials. So with that, I think I will let you know that this presentation will be available for order for about 6 months, and your questions will also get answered if I see them. So thank you all for joining us today. This is a great way to get information out there that I want out there in terms of what we can do about this field and what you can do to help us to make this field less of a problem and more of an opportunity, which I think is, as I said earlier, this is probably the opportunity of the ages. So finally, if you would like to join us on NBDA, here's our contact information, and you can go on our site and see some of the things I've talked about today. So again, thank you very much for your time, and I'll look forward to hearing from you if you have interest in talking about biomarker qualification.